Professor Asfar Azmi. Professor Azmi is Professor of Oncology at Wayne State University. He is leader of Molecular Therapeutics Program as well as Director of Pancreas Cancer Research at NCI Designated Comprehensive Cancer Center. He has made fundamental discoveries on role of aberrant nuclear protein transport in pancreatic cancer development and drug resistance. Today he is going to speak on targeting KRAS in cancer, emerging drugs and novel combination. Professor Azmi.
Thanks to the organizers for inviting me. So I'm associate professor, not professor yet, but anyhow. So these are my disclosures. I have no direct conflict of interest related to this work, and these are my funding. So let's talk about KRAS. This is a very interesting target. It's a high-value target in cancer because this is a protein that regulates several different processes, such as controlling critical pathways of cell division and growth. Now, how it works is it cycles between an inactive GDP-bound state and an active GTP-bound state. And this protein is important because of the functions it has, that when it's mutated, cells grow in an uncontrolled fashion. KRAS is considered to be like a brake in a car. So it tells us that if you want to stop the car, you put the brakes. And when the brakes go loose, then the car keeps on going uncontrolled. So this protein is highly valuable, as I said. It is this gene is mutated across all the cancers. You have about 30 to 50% of all cancers have mutation in KRAS. And in difficult to treat diseases like pancreatic cancer, we have more than 80% of mutations. And this is a big chart showing the different percentages across different tumor types. So we know all this about KRAS for the last 40 years, but the problem is that there were a lot of attempts made to discover drugs that faced a lot of challenges. And the reason for that is that KRAS is like a very circular protein, and it does not have a binding pocket for drugs to fit in. There is only one binding pocket, which is always attached to the GTP, and you cannot displace it with a small molecule inhibitor. And we know the challenges of targeting KRAS upstream and downstream, which have not been very effective. So all this changed a couple of years ago. There was a group, Kevin Shokat's group at UC Davis. What they found is that in the two conformations, the GDP bound and the GTP bound form, KRAS has an open and closed structure. So once it's in the GDP bound stage, there is a switch to region that opens up. It's shown here on the left side. And this conformation allowed the chemist to insert a small molecule drug. And you can see on the far right, this is a drug called AR-S853, which is neatly fitting into the switch to pocket of KRAS. So once this was discovered, there was a big race between several pharma companies to develop KRAS inhibitors. We published this review at that time. We called it the KRAS Game of Thrones because there were two main drugs, sotorasib and adagrasib, that were being developed simultaneously. And both had very similar profiles. And the reason why we called it a Game of Thrones is because if you look at the clinical data, the overall response rate of 43 and 41%, median progression-free survival or PFS of 6.5 and 6.3, they're almost mirroring each other. So this shows that the drugs which work in the same binding site with the same mechanism of action also show similar efficacy in the clinic. And after these two drugs, a number of other KRAS G12C inhibitors have emerged, and all of them are listed here. They are in various stages of clinical development. Some are in phase 1, phase 2, or phase 3. So among the latest drugs which have gone beyond sotorasib and adagrasib, drugs like the JAB21822 seems to be showing some better efficacy profile with an overall response rate of 57%. But if you look carefully at the data, we are not exceeding beyond 60%, which shows that almost in all cases there is resistance development to the KRAS inhibitors. And yes, that's the case. When patients were profiled after giving the KRAS inhibitors, there were many on-target, off-target, and lineage plasticity resistance observed. There were new mutations that were observed, and also these mutations were, in some cases, sensitive to a certain type of KRAS G12C inhibitor and not to the other type. So again, a lot of work is going on different clinicians are screening the patients who have undergone KRAS treatment, and we are identifying what are the causes of resistance. And that has led to a series of combination therapies that have emerged. Traditionally, people are trying to combine these drugs with chemotherapy, with immunotherapy, receptor tyrosine kinase inhibitors, and then some synthetic lethal interactions that I'm going to talk about. So our lab works on this protein called P21 activated kinases, and we did this synthetic lethal interaction analysis, which is a computational method, and we found that KRAS and PAC4 are synthetic lethal partners. And if you look at PAC4 function, it is a hub protein that links the KRAS to different downstream pathways. And if you have PAC4 overexpression, studies have shown that you have pronounced invasion, survival, cell proliferation, and so on. So what we did was we developed a drug-resistant cell, which one of the previous speakers had also spoke about. So we grew the G12C mutant cell lines in inhibitors like G12C inhibitors, MRTX849, over a long period of time, and we found that we could enhance the IC50. In fact, we had change of 42-fold IC50 when we developed the resistant cell lines. When we exposed these cells to the drug that inhibits PAC4, we saw a significant resensitization, and you can see that the IC50 is quite low. Then we did some combination studies. We took the PAC4 inhibitor, combined it with the G12C inhibitors, and this is you can look at the boxes in red where you see that the values are combination indexes. This is a very simplistic experiment where you combine two drugs. If the value is less than 1, that's called synergy, and you can see that when you combine the PAC4 inhibitor, that is KBT9274, with either sotorasib MRTX849 or AMG 510, which is, sotorasib is AMG 510 and adagrasib is MRTX849, you see a good synergy. We did several other follow-up assays like colony formation. You see the combination is more sensitive, makes the cells more sensitive. We did 3D culture assays. We found spheroid disruption. Now, historically, the first papers that published sotorasib and adagrasib data, they showed that you can block KRAS, you can block downstream effectors like phosphor, but then the phosphor tries to recover, and you can see in the box up there that at 72 or 48 hours there is recovery of ERG. However, when we tested our drug in combination with sotorasib or adagrasib, we found that there was a prolonged and persistent inhibition of phosphor, which is downstream of KRAS. So this shows that we can enhance the activity of these drugs by prolonging the downstream effectors inhibition. Then we next tested in animal models. Now, unlike the previous papers with the sotorasib or adagrasib, we have done 1/4 of the dose. We combined it with non-toxic doses of KBT9274, and you can see the lowermost blue-colored line, which shows that there is a superior inhibition of growth in these Miyapaka CDXs, which are KRAS G12C mutant tumors, and there was no body weight loss. When we followed up the residual tumors to look at the profile, we saw there was a persistent and long-term inhibition of phosphor in the tumors and other proliferation markers that are related to KRAS. We did a lung cancer model, which is a cell line derived model. We also saw similar results. Again, we are using 1/4 the dose, and you can see that there is a superior inhibition in the combo. And we did a prolonging of survival analysis. So we stopped the treatment after the 4 weeks or 40 days, and then we looked at the profile and we saw that there was enhancement in the survival of the combination group compared to single agent treatments or controls. So that was the story on PAC4. We are working on another combination, and this is regarding the nuclear transport system. So we know that proteins move between the nucleus and the cytosol in a gated manner, and there are some RAS-related proteins that help the movement, and that includes RCC1, RAN-GTP, and RAN-GTP. Now, earlier studies have shown that XPO1 or nuclear transporter protein XPO1 is a oncogenic dependency for KRAS mutant cancers. And why this is so? Because if you look at downstream of KRAS, below ERK, there are certain proteins like ribosome S6 kinase, which is known to facilitate the nuclear transport by activating the binding of RAN-GTP, RCC1, and XPO1 with the cargo. So based on that, there have been some drugs developed. We have worked on this drug for the last, I would say, 10, 12 years. This drug blocks the XPO1 function, retains the good proteins in the cellular compartment, and I don't know how I would play this video because I don't have a mouse here. But what we could see, and I think thanks for your help. So when we block the XPO1, you can retain the proteins in the nucleus of the compartment. So with this in hand, we combine the XPO1 inhibitors with KRAS G12C inhibitors. We have published this in Cancer Research Communications. We found that there was enhanced activity in terms of colony formation inhibition, 3D culture, spheroid inhibition, also enhancement in the survival of mice who were treated with the drug selinexor, which is a nuclear export inhibitor with sotorasib. And also many different models were used, whether it's MIA-PACA pancreatic or lung cancer model. So far I have told you about G12C inhibitor, but beyond G12C, G12D inhibitors have also emerged. Now this mutation is found in greater than 60% of pancreatic cancers. There is a new drug called MRTX1133 that specifically binds to it. And we have tested this drug in our lab. We see that some of the drugs we use, it gets synergized with the G12D inhibitor, whether it's colony, 3D culture, patient-derived spheroid models, and even in animal models, where we see superior inhibition of tumor growth. And if you look at the profile of these tumors or cell lines, we see that we could inhibit in a broad sense in the combination more protein tyrosine kinases, more threonine kinases, and we do many types of assays for this, such as the phosphocyte analysis using protein microarrays. And that shows that it's not just the cytotoxicity, but there is a molecular synergy when we combine the two drugs. So beyond these drugs, there are some RAS1 inhibitors which are emerging. Again, they have been evaluated in phase 1 trials. The previous speaker very elegantly presented these waterfall plots, and you can see that they are quite effective in blocking tumors. And then, aside from small molecule inhibitors, we have some degraders, we have antibody, we have vaccines, and we have some mRNA vaccines which are emerging and will be coming forward in the next few years. And IND-enabling studies or clinical trials are underway. So with that, my summary is that we knew about KRAS since 1982, but recently we got the KRAS G12C and G12D inhibitors which are emerging. Now we have more biologics also coming out targeting KRAS, but again, resistance to these drugs would be common, so the next phase of research would be developing rational combinations. So thanks for your attention, and I hope I can answer some questions.
Thank you very much, Dr.